Hello everyone, I welcome you all to Biology Lessons with Kalyani. In today's class, we will be starting with the new chapter, that is Plant Growth and Development. Here we will be discussing about different plant growth regulators or phytohormones, their physiological response. Okay, we will be discussing each in detail. So let's see the introduction part first. Now, plant growth regulators, in other words, the phytohormones, they are, phyto means plant, hormones means natural chemical messengers. So, they are the natural chemical messengers produced by plants which regulate different physiological activities in case of the plants. Clear? Now, these phytohormones, they are small molecules and they are produced by plants in very low concentration. The site in which they are produced, that is called as the synthesis site, and often it is seen that the phytohormones, they are translocated from the synthesis site to a different site where it has to show its physiological response, that is called as the target site. Okay, so phytohormones can be translocated to a target site in order to show its physiological response. Coming to the different examples of phytohormones, we have 5 main class of phytohormones, that is auxin, cytokinin, gibberellic acid, ethylene and abscisic acid. Now, these phytohormones, we can classify them into two types. How? On the basis of whether they positively impact or enhance a physiological response, that is, the phytohormone is said to act as a promoter, that means promoting a physiological response. Example of it can be auxin, cytokinin and gibberellic acid. Otherwise, a phytohormone can inhibit a particular physiological response and it will come under the inhibitor category. Example of it is abscisic acid, or in short ABA. Now, certain hormones also show both promoter and inhibitor activity. Example is ethylene. In the particular case of ethylene, we have inhibitory activity which is more than the promoter activity. Okay. Now, another thing is these phytohormones, they can show individual response, or they can also interact with other plant hormones, and they can interact in a positive way, which is called as the synergistic approach, or they can negate each other's physiological response and act as antagonistic. So this particular interaction within the plant growth regulators, it shows a overall physiological response in case of the plants. Is that clear? So this is the introduction part. Now let's see each hormone in detail. So we'll start with the auxin. Now auxin was discovered by Charles Darwin and Francis Darwin, the father-son duo, and the plant they used for the purpose was canary grass. So basically here the coleoptile, or the tip of the canary grass, they subjected it with the unilateral light. Unilateral light means in the same direction, single direction light was applied. Now what did they find out? They found that the coleoptile tip, it showed bending towards the light source. Okay. Then in the second phase, they tried to see whether if the tip is causing the bending. So what they tried to do, they removed the coleoptile tip of the canary grass, or they caused decapitation of the coleoptile. That is nothing but the removal of the coleoptile tip. And again they subjected it to the unilateral light. And the result was they did not see any bending or curvature of the coleoptile tip. Okay. In the next set of experiments, they tried to cover the coleoptile tip with an opaque cap or dark foil and covered the coleoptile. And again they subjected it to the unilateral light. and the result was no bending was observed. So what did they conclude from the experiment? They concluded that there is some transmissible substance. And where is it found? It is found in the coleoptile tip, and that diffuses in the presence of light to the shaded part, because bending will be always because of a overgrowth in the shaded region. Okay, so suppose we have two sides of a tip, and if this particular part is bending, that means there is more cell division which has occurred here. So they concluded that bending of the coleoptile tip is happening in the presence of the light, and the coleoptile tip has some transmittable substance which is causing that bending of the tip, and that transmissible substance is able to diffuse to the opposite side or the shaded side, so that there is extracell division or extracell growth and bending happen. Later on it was found that this transmissible substance is nothing but auxin. Okay. Now the bioassay or the extraction part, coming to that for auxin, it was performed in Avena sativa. Okay, so extraction of auxin, the discovery of auxin was done by Darwin, but the extraction of it was done by F.W. Wendt, and he took oats or the Avena sativa for his experimental purpose. Okay, oats or Avena sativa. Now auxin was also isolated from the human urine, because whatever we do not metabolize, it is always excreted in the form of urine. So what type of experiment did they perform? They also took a monocot, that is Avena sativa, and they took the coleoptile and they excised the tip, because it was clear that the tip is having something which is causing the bending. So they excised the tip and placed it on a agar medium. So what will happen? Whatever transmissible substance is present in the tip, that will diffuse to the agar. Next, they took the dark condition and then they placed the agar block which has the transmissible substance in one side, and again they repeated the experiment and they placed the agar block on the opposite side, and bending was found in both the cases. Hence it was concluded that the transmissible substance causing growth and bending, when they tried to isolate it, it was found to be auxin, and so the name auxin was also given by F.W. Wendt. Okay, so discovery of auxin, Darwin and Darwin. Plant taken, canary grass. Extraction of auxin, naming of auxin, it was given by F.W. Wendt. Okay, and the plant taken is oats or Avena sativa. Next, let's see the auxin physiological role. Now, if we see auxin, auxin is an indole derivative and the precursor for it is tryptophan. Now, the natural auxins, the example of it that is found naturally in case of the plant is indole acetic acid or IAA and indole butyric acid or IBA. Example of synthetic or man-made auxin is naphthalene acetic acid or NAA, 2,4-dichlorophenoxyacetic acid or 2,4-D, and 2,4,5-trichlorophenoxyacetic acid or 2,4,5-T in short. Okay. So next is, where is this auxin found? This auxin is produced by shoot and the root apex, and it migrates to the other parts where it shows the physiological response. And what are the types of the physiological response shown by auxin? The first one is apical dominance. Apical means, see, if you consider a plant, so we have two types of bud. One is the apical bud or apical meristem, another is the lateral meristem. So the phytohormone auxin, it induces the apical growth of the plant. That means the top part or the apical part of the plant undergoes more growth in the presence of auxin. Okay, so that is called as apical dominance. Now, you would have often seen that while hedge making, we trim the topmost part of the plant. Why? Because if we do so, the concentration of auxin will decrease, and as a result of this, instead of apical part, lateral part will grow, and we will have a bushy appearance for the plant. Okay, now this is also often used in pruning for tea or coffee plantation. That is why for hedge making, tea or coffee plantation, we perform the decapitation of the topmost part, or we try to trim the apical part of the plant. Okay. Next physiological action is auxin promotes cell division and cell growth. So its action is it increases the cell number, it increases the like cell, also undergoes an elongation purpose, and it acts synergistically with cytokinin, which is another plant hormone, and induces cell division in the callus. Okay. Next action is auxin. If it is a young fruit or leaf, it will prevent the abscission of the young fruit and leaf. But if it is old, we don't need it. That in that case, auxin will be performing abscission of the old plants and mature fruits. Okay, so it's a good hormone. Okay, now coming to senescence, which is getting old, auxin prevents senescence of the plants. Next action is xylem differentiation. Auxin, if you see, this is your leaf, this is your midvein, and you will have all the xylem and phloem here. So auxin helps in differentiation of this xylem. Okay. Commercially, auxin is also used as a weedicide. Example is 2,4-D and 2,4,5-D. The full form is given here, 2,4-dichlorophenoxyacetic acid and 2,4,5-trichlorophenoxyacetic acid. So both these 2,4-D and 2,4,5-T, they are used to control the broad leaf monocots. It is used to control only the broad leaf dicots, not the monocots. Okay, for controlling the population of the dicot, we use 2,4-D and 2,4,5-T. Okay. Next is like commercial application is parthenocarpy. Parthenocarpy means seedless fruits. Okay, auxins induce parthenocarpy in tomatoes, grapes, as well as cucumber. Also in tissue culture, auxin is used initially. Both auxin and cytokinin is used to get the callus from a plant part. Later on, from the callus, if we want to get root formation, we can use auxins. Also, if we have stem cuttings and we used to induce flowering in them, that time also we will be spraying it with auxin, and it will induce the rooting of the stem cuts. Okay. Auxin is an example of a feminizing hormone. It induces female flower production. It has been seen that in cucumber, ethylene majorly it will induce the production of the female flowers, but auxin also has a synergistic role in that. Okay, so that's all about auxin. Let's see the next plant hormone, that is gibberellins. Now, gibberellins, it was discovered by Kurosawa, and how he discovered is really interesting. So it was seen that in Japan, the rice plants, they were seen to be very tall, green, but later on they turned yellow also, but they were very tall, but they were not having any grains in them. So that is nothing, that disease is nothing but Bacane or the foolish rice disease. See, as a comparison, this is the rice plant which is suffering from Bacana disease, and the plants are abnormally tall and die without producing any grain, and this is a normal rice seedling. So this was uh, caused by the fungus Gibberella fujikuroe. Okay, now why it is causing this defect? So that's a fungus that is causing the yellowing, but that's Gibberella fujikuroe is also having a growth regulator, that is gibberellic acid, that is causing the rapid elongation of the length of the rice plants. Okay, later on, isolation of the gibberellic acid, it was performed by Yabuta and Sumuki. Coming to bioassay for gibberellins, it generally, as gibberellic acid, they are utilized to germinate the seeds. That is why the test we use to detect gibberellic acid is alpha amylase test. So before that, let's see the action of it. So two more things. Gibberellins are like chemically terpenes in nature, and the precursor for them is acetyl coenzyme A and mevalonic acid. The main function of gibberellic acid is they break the seed dormancy. So this is your dormant seed. Now we want it to be germinated. What should we spray? We should spray gibberellic acid. So what does this gibberellic acid do? It increases the alpha amylase activity, and thereby this increase in alpha amylase activity helps in the process of germination. Now there's another plant hormone, that is abscisic acid, which prevents like the seed to germinate. So both gibberellic acid and your abscisic acid, they are antagonists to each other. Okay, so now we saw that the main function of gibberellins is that it helps in breaking the seed dormancy. So the bioassay we perform to check the concentration of gibberellic acid is alpha amylase test. It is generally performed in barley. Okay, now more than 100 different types of gibberellins have been found, and the naming is done as GA1, GA2, GA3, and so on. So the like they are found in both the plants, and we saw a fungi also has it. So this is found both in plants as well as fungi. Now the first isolated gibberellic acid is GA3. Coming to the chemical nature of it, the name only suggests that they are acidic in nature. Let's get back to the physiological function again. So the first one is breaking seed dormancy. So second one is, if we spray the seeds with the gibberellic acid, it causes early seed maturation, and it is commercially performed in case of the coniferous plant. So coniferous plants spray gibberellic acid, there will be seed maturation. Third like activity of it is bolting in rosette plant. So what are rosette plants? The plants whose leaves are like that of a rose, that look like a rose. Like so, we can say cabbage, broccoli. Okay, so cabbage, broccoli, Chinese cabbage, beet, all these are examples of your rosette plants. So now, if gibberellic acid is sprayed on them, there is something called as bolting, which is nothing but the increase in the internodal distance. So see, this is the node where the leaf is attached. If because the internodal distance was less, so they were tightly packed and looking like a rose, or they were termed as rosette plants. When you apply gibberellic acid, this distance between the nodes, or the internodal distance, which was initially very small, it increases, and as a result of which the plant grows tall. If you would have seen something like we have broccoli, and the every all florets are tightly packed, there is something called broccoletti, in which they are separated from each other. That distance is a little more. So something like that. So bolting is because of the action of gibberellic acid. Next example, which is also utilized commercially, is increasing the stem length in sugarcane. So gibberellic acid, when you spray on the normal sugar cane, what happens? There is an increase in length. See, Bakane disease also length, they were abnormally tall. So that was not required in case of rice. We didn't get any grains. But here in sugar cane, where if the stem length is very like more, if the stem length is more, then definitely with the yield will be more. So 20 tons per acre yield was found to be increased with the spray of the gibberellin. Okay. Next physiological action is it increases the grape stalk length. So this is a bunch of grape. When you spray gibberellic acid, again the length is increased. Here the length of the stalk is increased. So in length of the stalk is increased, so more number of grapes can be found. So overall there is an increase in the yield. Also, this gibberellic acid delays senescence. Like old age, it is prevented or delayed because of the action of gibberellic acid. And as a result of this, the fruits also, the drop can be prevented when you spray gibberellic acid. They can be kept on the tree for a longer period, extending their market life. It has also been seen that in case of apples, when gibberellic acid is sprayed, the length of the apple increases. Even the quality or the shape, appearance also is enhanced as a result of that. Another commercial application of gibberellic acid is they are used in malting. Malting is used in case of your beverage production. We know all the alcoholic beverages, they are made from the grains. So when you apply the gibberellic acid, they increase the germination. So when the seeds are germinated in a shorter time, the malting process is enhanced. This activity is shown again due to the extended activity or more activity of the enzyme alpha amylase. Okay. So that's all about the hormone gibberellins. Let's see the next plant hormone, that is cytokinin. So here cyto means say cell, kinins means division. So cytokinins promote the cell division or cytokinesis. The plant hormones which are responsible for increasing or promoting the cell division, they are called as cytokinins. Now, these plant hormones, if we want to see their chemical nature, they are purine derivative, typically adenine. They are derivatives of the purine adenine. Then the discovery part, if you see, this cytokinins. Initially what happened, some experiments were done by Haberland in vascular tissue and later on performed in coconut milk. So what was it seen? That vascular tissue and coconut milk, they have some unknown substance. And this unknown substance is promoting the cell growth. Okay. Later on, Scooge performed repeated that experiment in vascular tissue and vascular tissue of the tobacco, also known as the tobacco pith culture, and he showed that the cytokinins, they are promoting the cell division in tobacco pith. Later, Miller he crystallized or isolated this a particular type of cytokinin, that is kinetin, which is not found in plants, but it was isolated from the autoclaved herring sperm DNA. So this kinetin was found to be 6-furfuryl aminopurine, and it was found to increase the cell division. Okay, so Scooge discovered cytokinins in tobacco pith culture, and Miller isolated the first cytokinin from herring sperm DNA, that was named kinetin. But it is important to note that kinetin is not found in plants. Okay, it causes similar effect in case of the plants, but as the natural cytokinins. But naturally occurring cytokinins, it was discovered by Miller in Zea mays or your maize plant, and this cytokinin was named as zeatin. This zeatin is also responsible for increasing the frequency of the cell division. Other sources of naturally occurring cytokinin are coconut endosperm. Okay, now the term cytokinin, it was given by Lentham. Okay, so this is all about the discovery of cytokinin. We saw the chemical natures, that is, it is purine derivative, typically adenine. Let's see where this cytokinin is produced. The cytokinin, as it induces cell division, where should be it found? It is found in rapidly dividing regions such as root tip, shoot apex, young fruits, okay, leaves. So these are the sites in which cytokinins are produced. Now, what are the physiological role of it? Cytokinins, they promote the cell division along with auxin. So they act synergistically with auxin and promote the cell division. Now, typically they are used in tissue culture experiments. Let's see how. We know tissue culture will take a part of the plant, and we will allow it to like culture in a solid medium. So this is the agar or the solid medium. Now, initially we took a control in which we did not add any auxin. We will just add cytokinin. We saw that there is no growth. That indicated that auxin and cytokinin needs to be present. Both should be present so that cell growth occurs. Okay. Then we took equal concentration of auxin as well as cytokinin. So what did we see? We see that there is undifferentiated mass of cell or the callus. So intermediate auxin and cytokinin concentration, we obtain your callus. Next, we tried to increase the concentration of auxin. So there it was found that the callus is developing the roots, or rooting of the callus takes place when auxin concentration is more than cytokinin. Next, we performed like experiment in which the cytokinin concentration was in more than the auxin. So in this case, it was found to be shooting of the callus. So so in tissue culture experiment, cytokinin initially helps in the formation of callus. Later, shooting of the callus can take place when the cytokinin concentration is more than that of the auxin. So that is an important physiological role. Next, we have something called the cytokinins. It helps in removing apical dominance, or it promotes the development of the lateral shoot. We saw in case of the auxin promote apical dominance. That is, it will make a plant taller. The top portion will grow more. But contrary to that, cytokinins will help in the lateral growth of the plants. That is, lateral buds are activated and branching takes place. Okay. Also, cytokinins, it promotes the nutrient mobilization in plants. All the nutrients can be used effectively. It delays the old age or senescence. It prevents the chlorophyll degradation. So cytokinins, it has a very important role in synthesis of chlorophyll. How can you remember it? C says cytokinin and C says chlorophyll. So both you can remember that way. So this cytokinin, it promotes new leaf formation and prevents the degradation of the chlorophyll. Also, it increases the shelf life of fruits as well as vegetables, because the cytokinins have a very positive impact in chlorophyll formation and protection. So for bioassay of cytokinins, we use chlorophyll preservation test or tobacco pith test. Is that clear? So that's all about cytokinin. Let's see the next phytohormone, that is ethylene. Ethylene is the only gaseous plant hormone. It was discovered by H.H. Cousins and it is synthesized in large amounts by tissues that undergo senescence. That is old plant tissues that produce large amount of ethylene. Also, the ripening fruits, that is the fruits that are getting ripe, they also produce large amount of ethylene. Okay. Now, the bioassay for ethylene is very important. That is called as the triple response test. Okay. Let's see what is triple response test. So, this is your control. That is no ethylene is added. This is a normal seedling and this is the length. This is the axis length. Okay. The seedling axis length. Now, when you apply the ethylene, what happens? The axis which is growing vertically, you see the seedling should be vertical in a control condition, that bends and there is horizontal growth. So that is the first response, horizontal growth of the seedling. Second is swelling of the axis. Swelling of the axis means this axis which is thin in the control, when ethylene is applied, what happens? The ethylene makes the axis thicker. Third response is apical hook formation. Apical hook formation is this topmost part or the apex of the seedling. It will make a hook like this, make a hook like structure. So these all tests, they were done in Arabidopsis thaliana, and this is called as a triple response test, which is widely used for bioassay of the ethylene. Okay. Now, typically this three responses, they are response to the stress. So what happens now in some conditions, the seedling, it encounters some stones or some hindrances. In that case, the hormone ethylene is released and it causes the seedling to grow sideways or horizontally, swells its axis and leads to the formation of the apical hook. So these all responses, they are a kind of stress response. Okay. Next, the physiological response of the ethylene. Ethylene promotes senescence and abscission of plant organs such as leaves and flowers. So it promotes the old age or the senescence, and also causes the fall of the leaves as well as the flower. Next, main physiological action of ethylene is in fruit ripening. It is highly effective in fruit ripening. How? It increases the respiratory or respiration rate in case of the plants. This increase in respiration rate is called as the respiratory climactic. It is the very important term. Okay. Now, because ethylene is very useful in the in fruit ripening, we mostly use aqueous solution of ethylene, uh, and it is sprayed on the unripe fruits. That aqueous solution of ethylene is called as ethiphon. This ethiphon causes a slow release of ethylene and helps in ripening of the fruits. This is the most widely used plant growth regulator or plant growth hormone. Okay, these all points are very important. What is the most importantly or most widely used plant growth regulator? Ethylene. Okay, ethylene is a gaseous hormone, but commercially ethylene is used as an aqueous suspension. What is it called? It is called as ethiphon. Okay. Now, we saw that it promotes abscission of the fruits and flowers in cotton, cherry and walnut. CCW, cotton, cherry, walnut. And this in cotton, it is also called as a thinning of cotton. Next physiological response is it in case of the rice plants, which is considered, this is a rice plant which is submerged this rice plant. So that's also a kind of stress. for the plant, right? Now the plant is submerged. How does it get like light and air? So what will happen under such stress condition? Ethylene is released and it causes both the internodal elongation as well as petiole elongation. What is internodal elongation? That means distance between the two nodes, or where the leaves are like leaves bud out, that is called as a node. Distance between the two nodes increases. Or also the petiole, the stalk, to where this is a stalk, right? This is your node, this is a leaf. So that distance, the petiole distance, also increases. So both internode elongation and petiole elongation, in case of submerged rice, it is because of ethylene, right? Don't get confused with gibberellic acid. Gibberellic acid causes generally a stem elongation, but for submerged rice, which is a stress condition for the rice plant, ethylene causes internode elongation and petiole elongation. Often this question is also asked. Okay, next let's come to the like action in case action of ethylene on seed and bud dormancy. Generally, the seed dormancy, it is broken by gibberellic acid, but ethylene also breaks the seed dormancy in some cases, such as peanut seeds. So peanut seeds, the seed dormancy is broken and germination is induced by the action of ethylene. Also, sprouting of potato tubers is with the action of ethylene. Ethylene also induces root and root hair formation and increases the surface absorption. Coming to flowering, it initiates flowering in pineapple and mango and increases the fruit set in case of the pineapple. Ethylene is a feminizing hormone. It also promotes the female flowers, typically in case of cucumber. Okay, so this is your female flowers. More female flowers are produced in cucumber, as a result of which the yield of cucumber increases. So this action, where else we have seen the feminizing hormone? If you remember, auxin also produces feminizing hormone here, but in case of cucumber, the female flowers are produced mainly by the action of ethylene, but auxin also plays a side role here. Okay, for fruit ripening, it's the main hormone. Also, it is used in for tomatoes and apples for fruit ripening in them. Okay, for ripening of tomatoes and apples, we use ethylene. Okay, so we saw ethylene is having both action as an inhibitor, because it is causing senescence, abscission, but it is also acting as a promoter. That is, it is inducing peanuts seed germination, potato tuber sprouting, right? So it is a hormone which is showing dual action. Let's see the next plant hormone, that is abscisic acid. Abscisic acid is a general inhibitor. It regulates abscission and seed dormancy. It is the inhibitor of the plant metabolism. It inhibits seed germination by counteracting the response of gibberellic acid. It is also a stress hormone. In case of water stress, that means where water is in deficit condition or water is less, okay, that time abscisic acid induces the closure of the stomata. We know stomata are the pores in the leaves of the plants which regulate gaseous and water exchange. So those pores are closed by the action of abscisic acid. So it is a hormone which helps in giving the drought resistance property to the plants. Okay, so it's a very important plant hormone which regulates the plant metabolism in extreme conditions. Okay, chemically, abscisic acid is a carotenoid derivative, precursor being xanthophyll. Coming to discovery part, 3 independent scientists discovered abscisic acid and termed abscissine dormin inhibin, but it was found to be the same compound, that is abscisic acid. The bioassay test for it is, there are 2 tests we have for abscisic acid: stomatal closure test, because it induces a closure of stomata, as well as alpha amylase inhibition test, because it also, abscisic acid counteracts action of gibberellic acid and reduces the activity or inhibits the activity of alpha amylase enzyme. What is this alpha amylase action? You know, alpha amylase is used in seed germination. As abscisic acid inhibits seed germination, alpha amylase inhibition test is also used for its bioassay. Okay. Next, abscisic acid plays a role in seed development, maturation, and dormancy. It acts as an antagonist to GA. Now, by inducing dormancy, abscisic acid, why do we need the seed to be dormant? Suppose the condition around the seed is not favorable and this seed tries to germinate, there will be seed desiccation. So, under such condition, abscisic acid causes seed dormancy, so that the seed is able to counteract the unfavorable condition. So see the physiological response like summarized here. It causes senescence, abscission of leaf and fruit, prevents seed germination, stomatal closure, and acts as a stress tolerant hormone for both biotic and abiotic stress. Okay. So let us summarize the different plant hormones here. Plants hormones, they are produced in different phases of growth and development. They can perform individualistic action, that is, they can have only single hormone can act for a physiological response, or they can act synergistically, like 2 or more hormones, they can combiningly produce a physiological response. Otherwise, this PGR can also be complementary or act in synchrony with each other, or positively impact each other, or they can also negatively impact each other. So let's see the common traits which are like found in plants. See, if you see seed and bud dormancy, we know that gibberellic acid helps us to overcome seed dormancy, and abscisic acid induces seed dormancy. So both are antagonistic to each other. Okay. Ethylene is also seen to overcome dormancy in some plants, peanut and potato tubers. Okay, but mainly gibberellic acid and abscisic acid, both are antagonistic to each other. Similarly, if you see abscissin, auxin, cytokinin, they prevent abscissin of both fruits and leaves, but abscisic acid it increases the abscissin, right? Also, ethylene plays in increasing the abscissin of the leaves and leaves and fruits. Coming to senescence, that is getting old, auxin, cytokinin, gibberellic acid, they prevent or delay senescence. Ethylene and abscisic acid, both they induce senescence. Next is apical dominance. See, auxin promotes apical dominance, and cytokinin, it is antagonistic and it overcomes apical dominance and helps in production of the lateral shoot. So in summary, all the different plant growth hormones, they act in different ways in different phases of growth and development of the plants. So with this, we complete the plant. So I hope you are clear about the different plant growth regulators or phytohormones. I will be also uploading a short video which will be summarizing the different discovery part, commercial applications, as well as different physiological role, which you can refer to after you refer this long video. And if you have any comments, queries, please comment. I will be very happy to help. And please, if you like the session today, please hit the like button and subscribe to my channel. Thank you everyone. Have a very good day.